So this mini lecture is going to be about the process of meiosis. And so I will walk you through the different stages that we see in our two cellular divisions of meiosis. And we'll talk about how the chromosomes segregate and the ways that we introduce genetic variability during this division process. And so if you recall, meiosis is a specific type of cell division that occurs in multicellular eukaryotes. This is the division that we use in order to be able to carry out sexual reproduction, right? We're talking about making the sperm and egg that we can use to make a new individual. And so because of that, the thing that we need to remember about meiosis is that it reduces our chromosome number by half. So typically what we're going to do is start with one diploid cell, meaning it has, two complements of our chromosomes. We've got homologous chromosomes. We're going to take that diploid cell, and through the process of meiosis and its two cellular divisions, we are going to create 4 haploid cells, cells that have one full complement of chromosomes and do not have homologous chromosomes. Again, because we're talking about meiosis in regards to sexual reproduction, this typically only happens for germ-line cells. So there are specific cells in your body that ultimately go through meiosis to give rise to sperm and egg. And it is only those germ-line cells that go through this meiotic process. And so I'm going to talk you through how that happens using our little model set that we work through in our group activity in class. And so again, I have my square cell here with my nucleus. And so for this particular exercise, just like we've done in the past, we are going to start with a diploid cell. Okay, so here is my diploid cell. This has 2 N chromosomes for a total of 4. So the black and red are a homologous pair. They represent the version from dad in red and the version from mom in black. And then we've got another pair of homologous chromosomes here. Again, green is from mom and orange is from dad. Now, before this diploid cell can go through meiosis, we do need to replicate our DNA. And so we need to make a copy of each of these chromosomes. So I'm going to go ahead and do that now. And so now I've replicated all of my DNA. I have 2 copies of both of the paternal chromosomes and 2 copies of both of my maternally derived chromosomes. And so now our cell is ready to actually enter into meiosis. And so here's what's going to happen. The first step of meiosis is to go through prophase 1. So if you recall, there are 2 major divisions to meiosis. Everything that happens in the first division gets the 1 at the end of it, and then when we get into the second division, there'll be a 2. So right now we're going into prophase 1. And here is what happens in prophase 1. We're going to have the degradation of the nuclear envelope. We are going to have condensation of our duplicated chromosomes, and we're going to go ahead and start to form our meiotic spindle. So in meiosis, there is no prometaphase, and so prophase 1 basically takes up the weight of what would have happened in the equivalent prometaphase of mitosis. And so we've got a lot that happens here in this very first step. We've got to get rid of our nuclear envelope, we've got to form our spindles, and then we'll talk about what happens to our chromosomes. So I'm going to go ahead and do these 2 things first. Again, fragmentation of our nuclear envelope, so we're just going to go ahead and get rid of a lot of this nuclear structure here. That way our microtubules will be able to go ahead and attach to our chromosomes when we need them to. So I've just fractioned my nuclear envelope. I also need to form my meiotic spindle. So I'm going to have my 2 centrosomes, which are going to create all of our microtubules. They're going to form, and they are going to start their migration to the opposing poles of my cell. Now here's the other things that happen in prophase 1 of meiosis. We have to start to deal with these duplicated chromosomes and how we are going to sort them so that they can be divided into our new cells. With prophase 1, it is not a matter of simply pairing up sister chromatids, but we also need to pair up our duplicated homologous chromosomes. Okay, so we need to find sister chromatids, and then we need to pair up our homologous chromosomes after that. And so that would look something like this. I'm going to start by pairing up my sister chromatids. So that means orange to orange, black to black, red to red, and green to green. But again, this is meiosis, and so it's not enough to just pair up our sister chromatids. Now I also need to align my homologous chromosomes, and that means to pair up the chromosomes I got from dad with those I got from mom. So I'm going to go ahead and do that in my cell. Okay. So this is what I mean by pairing up homologous chromosomes. I have sister chromatids, together we've paired the homologous chromosomes, the copies from mom and the copies from dad. Same thing for this set over here. I've got a pair of sister chromatids and a pair of sister chromatids, and I've arranged them as homologous chromosomes, the copies from mom and the copies from dad. So this happens in prophase 1. And that's because one of our big ways that we introduce genetic variability also happens here in prophase 1, and that is our crossing over event. Because these homologous chromosomes are close enough, we can actually get swapping of chromosome parts across homologous chromosomes. So what that means is this. What we want to pay most attention to, I'm going to separate these out just a little bit so you can see, is the pair of homologous chromosomes that are aligned right next to each other. So you've got a copy from mom and a copy from dad. There can be a break in the DNA in these two homologous chromosomes. And in order to fix it, we end up actually exchanging DNA from the maternal chromosome with DNA from the paternal chromosome. So what actually ends up happening is you get a crossing over event. You form these chiasmata, and then as that repair is fixed, we end up creating what are called recombinant chromosomes. And so I'm going to show you what that actually looks like. I'm going to take out these two that are recombining, and after that process is complete, what you're going to end up is with two new recombinant chromosomes. Notice now, again, this is a homologous pair, but they've swapped genetic information. This chromosome is now part mom and part dad. This one is majority dad and part mom. So these are your recombinant homologous chromosomes. They are still going to associate with their sister chromatid. But notice now I have new genetic options. This represents one type of genetic genome. This is a different genetic chromosome. And then these are each distinct genetic chromosomes. So we've introduced genetic variability. The same thing can happen with my homologous pair over here. I'm just going to separate them so you can see. These two homologous chromosomes that are aligned can go through this crossing over event, right? They can form chiasmata, and via breaking and rearranging the DNA, I can end up forming recombinant chromosomes that would look a little something like this. So you can see again, they've swapped DNA. I have my original copies that I made. This is, you know, the ones from mom and the ones from dad, but now I have these two new recombinant chromosomes. They're part DNA from mom and part DNA from dad. And so this is one of the big ways we can introduce variability. So again, we've got crossing over occurring here in prophase 1. Once we're done with all that crossing over, it's a simple matter of having the microtubules that are reaching out from our centrosomes attaching to the kinetochores that have formed on our paired homologous chromosomes. So we're going to have this pole will come and attach. This pole here will come and attach to the other half of our homologous pair. This pole will come and attach to this half of our homologous pair. And again, this one will attach. At that point, you enter into the next stage of meiosis, and that is metaphase 1. And this is where we see alignment, this time of our duplicated homologous chromosomes. Okay? So we're not going to align just sister chromatids. We need to align our entire homologous chromosomes. The sister chromatids of the pair are each attached to opposite poles. So one half of our homologous chromosomes will go to this pole, and the other half of our homologous chromosomes will go to this pole. Okay? And then I'm going to talk to you as I show you that this is where independent assortment happens, right? So we talk about this in class. Independent assortment is another way that we can introduce genetic variability. So let me show you what I mean by all this. So again, our microtubules have reached out and attached to our kinetochores, and they're going to start tugging and pulling these back and forth until they align up at the metaphase plate. Same thing over here, tugging and pulling until we align up at the metaphase plate. So now we have all of our chromosomes aligned at the metaphase plate. Again, we're aligning by homologous chromosomes. I'm not using just a pair of sister chromatids to align. I'm aligning all of my homologous chromosomes together. All of my homologous chromosomes together. One half of this, so this side, will attach to my left pole. Over here, the other half of my homologous duplicated set will attach to the right pole. Same thing up here. These will attach to the right, and these will attach to the left. Now, how does independent assortment come in? Independent assortment refers to which half of the homologous pair is attached to which pole. And so I aligned the first time my chromosomes up this way, such that these ones that are mainly dads go to the right, and these ones that are mainly moms go to the left. But I could have just as easily switched it and had them align up this way so that moms go to the right and dads go to the left. It doesn't matter. You have a 50-50 shot which orientation those chromosomes decide to line up in. The same thing happens over here. You have a 50-50 shot of either having this arrangement or having this arrangement. It doesn't matter. And even more importantly, the way that this homologous pair orients has absolutely zero influence on how this homologous pair orients. They do not dictate what other homologous chromosomes do. So I can orient this one anyway, and I can orient this one anyway, completely independent of what this chromosome is doing. Hence the term independent assortment. Once we have aligned our chromosomes at the metaphase plate via independent assortment, now we can enter into anaphase 1. And this is where our homologous chromosomes are going to separate and migrate to the opposite poles. During this process, we are only splitting homologous chromosomes. We need to make sure our sister chromatids, our exact duplicates, remain together. So that will look something like this. This pair will migrate this way, and this pair will migrate this way. So I've separated the homologous chromosomes, but I'm keeping my sister chromatids together. Same thing up here. They're going to migrate to the left, and they will migrate to the right. I've separated my homologous chromosomes, mom to one direction, dad to another, but I've kept my sister chromatid pairs together. So now I'm going to go into telophase 1. Basically, I'm going to reform some nuclei around these segregated chromosomes. But because of how I split my homologous pairs, I am now, for the first time, forming a haploid cell. Each cell is only getting one of each set of homologous chromosomes. I'm either going to get the big chromosome from mom, or I'm getting the big chromosome from dad. I am not getting both. Same thing for my shorter chromosomes. I'm either getting the one from mom or the one from dad. My nucleus is not going to get both, and so I'm really only ending up with one set of duplicated chromosomes. That is going to make these haploid cells. Okay? Again, they're still in their duplicated state. They're still sister chromatids, but these are the same chromosome, and it's just one version. They are no longer homologous. So let's go ahead. We're going to, dissolve our mitotic spindle, and we're going to create our new nuclei. So let me get my cells here get my chromosomes into my new nuclei. Okay, so here I've got my two haploid cells that have formed at the end of telophase 1. I've got sister chromatids, but only one set of chromosomes, one large and one small. Same thing over here. So these are haploid cells. They're now going to enter into the second division of meiosis. And so the first stage of that is prophase 2. And it goes through pretty much the same process. We're going to disintegrate the nuclear envelope. We're going to go ahead and create new mitotic spindles. So I'll go ahead and do that for each of my cells here. Let me see if I can zoom out a little so you can see everything. All right, so we've got new mitotic spindles for each cell. They're going to start to go ahead and form our microtubules so we can attach to our sister chromatids. I want to stress at this point in prophase 2, we no longer are going to see crossing over. These duplicated sister chromatids are not going to cross over with each other. That event only happens in prophase 1. And so really at this point in the stage 2 of meiosis, it's just about going through and separating out these paired sister chromatids. So no crossing over in prophase 2. Once our microtubules reach out and touch our kinetochores along our sister chromatids here in prophase 1, then we can go into, or prophase 2, then we can go into metaphase 2. This is where we're going to get alignment of our sister chromatids now along that metaphase plate. And each half of the chromatid will be attached to a different pole. So I'm going to go ahead and do that for both of my cells. Right? So you've got the tugging back and forth until you align your sister chromatids up on the metaphase plate. There we go. Okay, so we've got, again, sister chromatids aligned along the metaphase plate in each haploid cell. The chromosomes, or the half of the sister chromatids on the right, will connect to the mitotic spindle on the right. Those that are aligned on the left in both cells are going to attach to the left mitotic spindle. So now, as we go into anaphase 2, our sister chromatids are going to be separated. So that cohesin that's been holding these sister chromatids together is going to get cut and allow the chromatids to separate. Now we are going to see these sister chromatids for the first time become individual chromosomes. And those individual chromosomes are going to migrate to their opposing poles. So that would look something like this. All right, so I'm cutting my cohesin. So now those microtubules can shorten and drag my chromosomes to the opposite pole. So I've separated my sister chromatids. These are now individual chromosomes. Okay. So then the last phase is just to go through telophase 2. We're going to regenerate the nuclear envelope and form the new nuclei around each of these 4 daughter cells. and we're going to go ahead and complete cytokinesis. So that by the end of this, at the end of this second major division of meiosis, we've created 4 haploid nuclei, each one of which is genetically different from each other. So let me bring in some new nuclei here. Right, so we've got dissolution of our mitotic spindles. We no longer need to pull our chromosomes apart. So these 4 will stay in that cell. We're going to move these into a new cell that forms. And these 2 will stay here. If I can get them all. So again, I've got 1, 2, 3, 4 new daughter cells. Each of them are haploid because they have only 1 copy of each chromosome. They either got mom's or dad's or one of those recombinant versions. They did not get 2. If you remember at the beginning, we started with a cell that had 4 chromosomes. These are haploid. They all only have 2. They are genetically different. Here, I got the big chromosome from mom and the little chromosome from dad. Down here, I have my big paternal chromosome and a little maternal chromosome. Based on how I align them during metaphase 1, this particular cell is getting recombinant chromosomes. So it's half dad and half mom for each of them. Same thing up here. I could have gotten a completely different result in these 4 haploid cells if I had arranged my homologous pairs differently during that metaphase 1. And so meiosis, again, you're going to basically have your chromosome number in each cell that results. You create 4 new haploid daughter cells, each one of which has a completely different genetic makeup relative to the other one. And so that is the process of meiosis. If you're still struggling, I recommend you watch this again and try to go through the process with me using some pairs of your socks, because I think physical manipulation will help. And if you still have questions, please come see me.